In this activity, we're going to examine cells from a potato tuber. Potatoes are not roots. They are underground stems called tubers that are specialized for storage. The cells you'll observe here are very different from leaf tissues like onion or Elodea because their primary role is storing energy for the plant. Potato tubers are made up mostly of parenchyma cells, which are large, loosely arranged cells designed for storage. Unlike leaf tissues, these cells are not tightly packed and are not involved in photosynthesis. Their main job is to store energy that the plant can use later for growth and development. Inside potato cells, you'll find specialized plastids called leucoplasts, which are used for storage. A specific type of leucoplast, called an amyloplast, stores starch. When potato tissue is stained with Lugol's solution, the starch inside the amyloplasts turns dark purple, making these structures much easier to see under the microscope. Before we look at the microscope images, pause and answer this pre-lab question in your lab notebook. What substance would you expect to find stored inside a potato cells, and why would this be important for the plant? Think about what plants store for energy and why having a storage organ like a tuber would be beneficial for survival. Go ahead and pause now, then return when you're ready. Here's what potato tuber tissue looks like under high power, where individual potato cells are easier to examine. As you look at this image, start asking yourself, where are the boundaries of each cell? Do you see dark purple structures inside the cells? Are the cells tightly packed, or are there clear spaces between them? Now let's confirm what you're seeing. The cell wall outlines the boundary of each potato cell and provides structural support. The small dark purple oval structures inside the cells are amyloplasts. These are starch-storing plastids that become visible when stained with Lugol's solution. You may also notice intercellular spaces, clear gaps between cells. These occur because the cells are not tightly packed and allow for gas exchange within the tissue. These features all reflect the potato's role as a storage organ. In your lab notebook, draw 2 to 3 potato tuber cells based on what you observed. Be sure to include and label the cell wall and amyloplasts. Focus on accurately representing the spacing between cells and the abundance of amyloplasts rather than artistic detail. Pause the video here and resume when you're finished. Now compare the potato tuber cells to the onion epidermal cells you observed earlier. How do their shapes differ? What differences do you notice in how tightly the cells are packed? Think about how these differences support the function of each tissue. Storage in potatoes versus protection in onion epidermis. Make sure you pause this video to answer the question in your notebook. Finally, think about how potato tuber cells compare with Elodea leaf cells. Why is the central vacuole more prominent in Elodea cells? Why might it appear smaller or less obvious in potato cells? Consider the different roles these tissues play in water balance, photosynthesis, and energy storage. Take a moment to pause the video to answer the question in your notebook. This activity highlights how plant cells are specialized for different roles. Potato tuber cells are designed for carbohydrate or food storage, which is reflected in their large size, loose arrangement, and abundance of amyloplasts. As we move forward, you'll continue to see how plant cell structure supports the function of the tissue and the needs of the plant.